start with a tiny request for the audience. Please close your eyes. Truly do that. Imagine yourself going to the lab and starting to setting up an experiment. And then you think about what a tedious and long day it's going to be because you need to perform the experiment and then to do additional steps in order to see your data, to observe it, and to analyze it. And then imagine yourself just reaching out to a drawer, open it up, put in a glass, and wearing it, and just observe simply by that with the result, you see the result in your sample. Now, open your eyes, please, and let me introduce you to a promising technique that will give you this ability, this amazing ability, in nanoscale. We'll do that not only in solution, but also in living cells. And this is what is the most amazing thing in this method, and it's called in-cell EPR. Deciphering protein dynamics and structure in their physiological milieu, the cell, it's one of the main goals in structural biology in recent years. The native environment of the protein, which co-involve with the cell component, may affect significantly on the protein properties. However, the majority of protein studies had been conducted under artificial laboratory settings, and we can see that on the left panel. However, these settings are significantly differ from the cellular environment, which is complex and completely different. Therefore, many efforts had been made in order to adjust the biophysical methods to the cellular environment. And this, however, presents the biophysical method with great challenges. And therefore, this field is still far away from establishment. And this is where we are coming into the story. Among these techniques, electron paramagnetic resonance, or shortly EPR, and remember this word EPR, has been shown to be potentially a promising tool in order to investigate the protein inside living cells. Using this technique, we can probe the protein dynamic and structure in a nanoscale resolution. So it's basically where all the action is happening if we're talking about biomolecules. It is highly selective, which means that it is powerful for in-cell studies. It is not destructive, so we do not destruct either the protein or the cells. And it's applicable to any protein or complexes that we can think of, so we do not have any limitation either on the protein type or on the size of the protein. And why is it so powerful comparing to the other techniques? Because an inherent requirement of this technique is to spin label the protein, and then we can solely track the spin label protein, which is again special probes that bears magnetic features, and we can track that upon magnetic field among all the other cell components that are mostly diamagnetic, and therefore we are basically blind to them. And how do we do that? First, we are expressing the protein in cells and purifying it. And until here, everybody knows how to do that well. I don't need to explain. Next, we are performing an spin labeling of the protein, and we can use many different spin labels. It depends on the method that you want to use or the information that you want to obtain. And then we deliver it into the cell, and we are performing that commonly using electroporation. Now, some people might say, what? Yes, we are using electroporation in order to reintroduce the spin-labeled protein, and then we can perform our measurement and obtain either the structure or the dynamics, and I'm going to explain it a bit better next. Since we can incorporate our protein into mammalian or bacterial cells, we can explore a wide range of protein using these techniques. Again, as I promised you, I will explain just a bit about the techniques that we are using. Using suitable spin labels, we can either detect the local dynamic of the position that we locate our spin label, and this method called X-band continuous wave, or in shortly, CW-EPR. Basically, just in a nutshell, we are following the line shape from a characteristic one. Here you see these spikes that is characteristic for a fast motion, and as they go broadening, then we see the mobility is getting restricted. We can also probe the distance distribution between two spin labels that we located, again, on the protein at strategically position that we chose in advance. And this using a pulse-dipolar EPR, or more specific, double electron-electron resonance, or shortly DEAR. And basically, again, from here we can extract the distance distribution. So it's either if you have a change in conformation, you can see the change, and also you can see the population between each conformale. And that is amazing. Now, from here, I would like to show you an example for the application of this method. And in this study, we reveal the dual behavior of PPIB in solution and in cell. The first thing that we wanted to understand is the dynamics of the protein. The protein is an E. coli chaperone, so it's very important for the viability of the E. coli. And here we measure it. We acquire X-band CW in solution, which it is represented by the dotted line, and the solid line represent the spectrum acquired once we incorporate it into our cells. And surprisingly, each variant that we chose, either the single label or the double label that you can see here, or with different spin label, it always showed this reduced from this change from a relatively spiky spectrum, which show a high mobility, to a low mobility with this broadened line. So from here we can say that in both locations that we decided to explore, we see that the dynamics is getting reduced once it goes into the cell. And that makes sense because of the complicated and compact and complex environment. Additional thing that we wanted to see is the distance distribution. So we incorporate double-labeled protein. We deliver it into E. coli cell, which is the native environment of our protein, into non-native cellular environment, which is HeLa cells. They represent the mammalian cells. And we compare it to in solution. If you can please look on the panel C, you can observe that only in the native environment of our protein, you can see this broadening that represents an expansion of the conformational space, which basically means there is more conformers because our protein is interacting with the cell component that is his binding partners, basically. And this is a great example for the scope of biological question that we can address using this technique, and the potential that the in-cell EPR holds with it, either as a standalone technique or as complementary to others. And with that, I will thank firstly for my supervisor, Daniela Gorfab, and for the other people who contribute, especially you have Barak, to this work, and for you for listening, and I hope you enjoy this deep dive into the EPR world. We have